It is now my pleasure to introduce Barbara Kinner, who will moderate our scientific panel. Barbara is the founder of the Kinner Family Research Fund based here in the United States. She's also a founding member of the World Pancreatic Cancer Coalition and sits on the steering committee. Barbara, over to you.
Thank you, Julie, for that welcome. I'm honored to have this opportunity today to facilitate what promises to be a very valuable scientific session. The specific topics that we're going to discuss today were selected as areas of high interest in a survey completed this past summer by all of you members of the WPCC membership. In line with this, we're going to first discuss the promises and challenges in early detection, including the roles of genetic testing, risk stratification, and imaging. And subsequently, we'll learn what innovations are on the horizon. I'd like to take a moment to introduce our panelists who are globally well respected within the field of pancreatic cancer. They are recognized for their leadership, passion, determination, motivation, and collaborative efforts to improve survival for patients with pancreatic cancer. Their full bios are included in your meeting packet. But briefly, Dr. Elizabeth Liz Jaffee is an internationally recognized expert in cancer immunology. She is Deputy Director of the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins, co-director of the Skip Viraj Pancreatic Cancer Center, and associate director of the Bloomberg Kimmel Institute for Cancer Immunotherapy. Her research focus is on developing novel immunotherapies for the treatment and prevention of pancreatic cancer, and she has served on a number of committees at the National Cancer Institute, including the co-chair of the Biden Moonshot Blue Ribbon Panel, which identified high-impact research priorities. She currently serves as Chief Medical Advisor to the Lustgarten Foundation for Pancreatic Cancer Research. Welcome, Liz.
Thank you.
Dr. Anirban Maitra is a professor of translational molecular pathology and scientific director of the Sheikh Ahmed Center for Pancreatic Cancer Research at the University of Texas MD Anderson Cancer Center in Houston. He has been a champion of rational early detection of pancreatic cancer, specifically geared towards high-risk cohorts. In this regard, he currently co-leads a United States national effort to recruit patients with new onset of diabetes who have been identified as high-risk cohort for this disease. He is also the principal investigator of the NCI-funded Early Detection and Research Network Clinical Validation Center focused on pancreatic cancer early detection and collaboration. And welcome, Anirban.
Barbara, thank you so much.
I'm going to spend about 45 minutes asking our panelists questions. I've been given permission by Liz and Anirban to be informal and call them by their first names. We'll then take audience questions via the Q&A window. If you see up on the right-hand side, you have chat, then you have participants, and then you have Q&A. So any questions that you might have, please feel free to put them in there while we're talking, so that when it comes to the Q&A session, that we'll have them prepared. There will be a video recording as well as a summary sheet of the main points from this session, and that'll subsequently be made available to all WPCC members via a link. So if we think about current important efforts in early detection, and we're going to hear a lot of what is going on, but as a context for this discussion, we need to remember that a majority of pancreatic cancers, about 90%, is sporadic. They occur without a family history or a genetic predisposition of the disease. It is estimated that it takes more than 10 years before an initiating mutation in a normal pancreas cell progresses to the non-metastatic parental founder cell of pancreatic cancer. And at least 5 more years are required for acquisition of metastatic ability. This window of time offers an opportunity for curative interventions if the disease could only be diagnosed at an earlier stage. Recent evidence also supports the hypothesis that once pancreatic cancer becomes detectable, the clinical progression from early stage to advanced stage disease is rapid. Therefore, the best outcomes will be tied with detection at the earliest stages of the disease. However, all of us are here that we are keenly aware that most pancreatic cancer patients globally are diagnosed late stage when they are no longer eligible for surgery, making the disease difficult to treat successfully. Anirban, I'm going to ask you this question. And then Liz, as we talked before, just jump in whenever you'd like to. Who is currently considered at high risk for pancreatic cancer?
Well, thank you so much, Barbara, for having this session. I just want to say, first of all, thank you for the welcome packet to the WCC. Very much here. I'm having my my butterfly pea flower de-stress tea with me here. So if you see me not stressed at all, it's because of the tea that I'm having. So thank you so much for that. So I think it's a very important question because, as you know, in 2019, the USPSTF, which is the main body that's charged with giving evidence-based guidelines for screening in the population, specifically gave a failing grade of D, which is do not screen to the general population screening initiatives. In fact, they typically do this when they think that the potential for harm outweighs any potential benefits. So that means that in the context of pancreatic cancer, when we talk about screening and surveillance programs, we are talking about certain very predefined high-risk cohorts. So what are those high-risk cohorts? Well, the two that are probably the most well-established and work done by many folks around the country and actually at Hopkins, to be honest, a lot of pioneering work came out of the group at Hopkins. The first one is obviously patients who have a family history or carry deleterious germline mutations. About 10% of pancreatic cancers, give or take, arise in the backdrop of these germline mutations, and the most common of that is BRCA2. And a second common player that has emerged in the last few years, again, the discovery was made at Johns Hopkins, is a gene called ATM. So between BRCA and ATM, those two together account for the most two common known causes of germline mutations. And then there are less common alterations as well, like P16 and the Lynch syndrome genes, etc. So that's the one big group. And some patients or some families, we really don't know what the mutation is, except that they have multiple family members with pancreatic cancer, and that those families are often called familial pancreatic cancer families, where we don't really know what the mutation is, except we know that there is definitely a predisposition, and that's why you have so many members with this disease. So that's a big sort of high-risk cohort, and the USPSTF actually specifically excludes its do not screen recommendation for patients who have, or individuals, I should say, who have a familial predisposition. And in fact, centers around the country, including MD Anderson, Johns Hopkins, and all these other academic centers, have surveillance programs for such high-risk individuals. Mm-hmm. The other high-risk cohort are patients who have pancreatic cysts. So these are these fluid-filled sacs, and they have very sort of big names. Intraductal papillary mucinous neoplasms, or IPMNs, is the most common subtype. And we know that a minor subset of them, but a subset definitely, goes on to develop pancreatic cancer over time. And so because these cysts can be picked up on imaging studies, patients who have such cystic lesions are followed over time for progression to adenocarcinoma. Now, I will emphasize that the overwhelming majority of patients will actually not develop cancer in the context of cysts. There are about, by some estimates, 6 million Americans who have a silent cyst in their pancreas, and about 15% of pancreatic cancers in a year arise in the background of cystic lesions. So let's say 55, 60,000, we're talking about 10,000 patients where the cancer arose in the backdrop of a cyst. So again, there's a huge number of individuals with cysts, but only a small slice of those will go on to develop cancer. And in fact, there's a lot of research going on again at centers like Anderson and Hopkins, I tried to identify who are those or which are those bad actor cysts that will progress to cancer versus the ones that are innocuous. And the third slice, so we talked about familial history, we talked about cystic lesions. The third slice, which is not a risk factor in the true sense, when we talk about risk factors, we talk about future risk of cancer. So somebody who has a BRCA mutation has a 3 times greater lifetime risk of pancreatic cancer than somebody who doesn't have that. The third category, which is new onset diabetes, is a special category of risk in the sense in patients where this is caused by a pancreatic cancer, the cancer is already there. So I want to make sure that the audience understands the difference. Long-term diabetes over many, many years, 10, 20 years, is a very modest risk factor, about 1.5-fold or 2-fold at the most for pancreatic cancer. New onset diabetes is not a risk factor in the way we think of risk factors, but is a manifestation or a symptom of a cancer that's there already, but has not presented itself with classic findings like jaundice, weight loss, abdominal pain. Why is this important? This is important because if we can identify that tiny sliver of new onset diabetes patients where the cancer is causing the diabetes, we might be able to get that cancer out at an earlier stage than if we had waited for the usual manifestations of cancer to appear, such as jaundice, weight loss, etc. So again, it's a risk factor, but it's not a risk factor in the traditional sense. It's basically a symptom of a cancer that's already there. Okay? So those are the 3 major categories.
Yeah. And we often think about some like modifiable risk factors, like smoking, alcohol, diet, obesity. Inflammation is one that's coming up that there's a lot of work in it. Liz, can you comment a little bit about that?
Yeah, I think that's a great question. We don't know yet how inflammation plays into pancreatic cancer development. We certainly know once you have your initial genetic mutation, KRAS, which is really all of these different premalignant lesions typically start with the mutation in one gene. Now, the mutations may be different, but this one gene is so critical to this cancer, which sounds bad, but actually it's a good cancer for trying to intercept, because you have to target this one pathway initially. But that one gene can cause so much change in the organ it's developing in, which is the pancreas, and it sets up inflammation. Now, initially, the immune system is probably saying, hey, there's this big change like a virus. Let's come in. All the cells that would normally react right away, these monocytes, these even T cells, B cells, they come in. They want to do something about what looks like an insult or a new change in this organ. But what happens is that first mutation starts to set up pathways that really take advantage of these cells and get these cells to turn on the environment in favor of the cancer's growth. And what does it do? It excludes a good immune response, because these cells, instead of causing what would be a good immune response, they now cause more inflammation of the organ that inhibits an anti-cancer response, but it's great for the cancer to grow. Just building on what Anirban said about, screening, I'd like to think about this more globally. When I first got into this field, I was really excited that the immune system had such great power against infection. And where is its greatest power? It's in prevention, right? Not interception, in prevention, right? So we prevent polio. Now we're preventing COVID. We all know a lot more about vaccine development to prevent COVID. Well, you can think of a cancer as something very similar to a viral infection, because viruses get into cells. And then in the cells, they make these proteins. They cause the normal cell to make proteins that are viral proteins to help it spread. Well, that's what happens in a cancer cell. You have these mutations in a normal cell that helps it grow and spread. And so the immune system can see a cancer that has these genetic changes in the same way it can see a virally infected cell. So we're able to develop similar types of vaccines, hopefully to intercept. And we have examples of this. We have the human papillomavirus vaccine, We know that if we give that vaccine to kids who are 10 to 15 before they become exposed to the human papillomavirus, we know we can prevent them from getting the virus and from getting the diseases associated with the virus. Well, yes, we can think about screening people who are at high risk, but if we had a safe enough vaccine, this is a bad cancer. This is a deadly cancer. Why wouldn't we want to just give a vaccine to prevent the cancer like we do against human papillomavirus associated cancers? So that's my big dream, is to be able to take these vaccines off the shelf and really vaccinate young adults to prevent all sorts of cancers. I think pancreatic cancer is one that if we put enough effort into, it could be a model for this, for a non-virally associated cancer.
Thank you. That is really interesting to hear about that. There's also some, I want you to just talk a little bit more about this area, because there's some non-modifiable factors also, like we know age, for instance, is one that it typically is older. Well, I have another question on that in a second. We also know that people with Lynch syndrome, Ashkenazi Jewish ancestry in the Black community have a higher incident rate. How is that being addressed in research at this point?
Liz, did you want to take that, or?
Yeah, I mean,
I'm happy to take it too.
There's a lot of research ongoing. I think, there's been years of research, and if I were sitting in this audience, I'd say, yeah, but you haven't really done a lot for this disease, and you're absolutely correct. We have not done enough for this disease. But what may not be obvious is that we now, for the first time in the past 4 or 5 years, have technologies that allow us to really get at molecular differences, molecular changes that result as a tumor is transforming, becoming a tumor, and then metastasizing. And it's allowing us to better understand, in association with BRCA, with all the different genetic alterations that we see. And so we also can look at this if we have the enrollment of a diversity of patients. We can look at this among different cultures, different ethnicities, different races, and be able to look at why there are these differences. I know that these occur, but we still don't have the biospecimens in trials to be able to really discern big differences of why certain groups get what they do. I don't know if that answers your question. Maybe Anirban has a better answer.
No, I'd say this is such an important point, Liz, what you just raised, which is that so many of our studies unfortunately lack the diversity that represents the reality of where pancreatic cancer happens. I mean, we know that African Americans, for example, have both a higher incidence and unfortunately worse outcomes for this disease. Some of it is biological, some of it is structural. In terms of the inequities that exist in society. And we have to work our way through and understand, what can we address? What are the addressable parts of this that we can change? Just to give you an example, I was shocked because I recently wrote an NIH grant on this, looking at increasing the diversity of patients who undergo biomarker studies, because obviously a big interest in my lab is early detection and biomarkers. The diversity in many of the national biobanks for pancreatic cancer is abysmal. I mean, we're looking at numbers of less than 5%, 10%. The TCGA, which was a giant sequencing effort in pancreatic cancer that was funded through the NIH, unfortunately barely had any African American patients. And so I think addressing the issue of diversity is one of critical importance. And one that I think, and Liz is closer to the NCI than I am, but I think the NCI takes this very seriously and one that is really trying to address in a systematic way.
It was a major recommendation of our Blue Ribbon panel that we really need to get at two very important things. One is differences in cancer, outcomes of people of different backgrounds, economic, socioeconomic, cultural, racial. But the other thing is dissemination. And as you said it, Anirban, you're absolutely correct. We are not doing a good job of getting to the people who really have no access right now. And we need to encourage people to come on these trials, but how do we get these trials out to them? They don't have transportation. They don't have the money to travel. They don't have health care that's good enough for this. It's really, as a physician, it's very frustrating and difficult. And then, I'm just talking about the US, I know that the best, but I could tell you at least once or twice a week, I get emails from people around the world, and they have even less access.
Mhm.
So it's a real problem.
Exactly.
We'll talk a little bit later about maybe how some of that's being addressed at this point in time. It was interesting hopping around tables before we went into this session. Is this exact question came up, is that although pancreatic cancer is generally viewed as a disease for those who are older, there's growing evidence of an increase in pancreatic cancer amongst younger patients. What do you feel is driving this?
Well, I can take that. I mean, I think it's a combination, as with everything else. As I say, the answer, it's complicated. It's several things together. Number one, I think that this is a disease of the median age of this disease still remains 69. Most patients are still in the elderly age group, but you're correct that we are seeing more younger individuals. Some of it is driven by the so-called, diabetes and obesity epidemic. Unfortunately, there's a lot of obesity now that starts early in childhood. Unfortunately, especially in developed nations, and that's where I think we sort of program individuals to a lifetime risk of not only pancreatic cancer but other cancers as well. One of the cancers, for example, that's going up in numbers is endometrial cancer, and that's one disease that we know has a tremendous relationship to high BMI. So again, we are seeing a lot of folks who unfortunately have had obesity for many years starting in childhood. That's number one. Number two, honestly, we've also gotten better at diagnostics. I mean, many of these numbers come from national registries where maybe 20 years, 30 years ago, before molecular profiling was rampant, something would be just called disseminated cancer, not otherwise specified. Now we actually can work up these cancers, these what we call cancers of unknown primary, and using molecular data, figure out, well, it's actually coming from the pancreas. And so it's coded as a pancreas cancer in repositories. With all of this said, there's also a greater awareness now about the importance of high-risk cohorts and individuals in families where there is cancer are often getting self-tested. When I was at Hopkins, this, I mean, to test BRCA used to cost thousands of dollars with one company. It was $5,000 to sequence one gene. Now you can get multi-genes tested for $250. It's very easy. And so and plus you have all these other direct-to-consumer tests as well. So a lot of folks are getting tested. They're finding out they have a germline mutation that might predispose them to cancer, pancreatic or some other cancers, undergoing surveillance, and early cancers are being found, which is a great thing. I'm delighted that we're finding these early cancers, but it's also adding to the numbers. In some registries, for example, at Johns Hopkins or at MD Anderson, especially in younger individuals, those who have cancer under 50, pancreatic cancer under 50, 1 in 5 or 20% have a pathogenic germline mutation in the under 50 cancers that are diagnosed. This was our data, and I think Hopkins and others are pretty similar.
I'll add one more thing to this. It's a really important question. I think Anirban raised a lot of the factors that may play into these earlier pancreatic cancers. We're trying to study this from a kind of taking an out-of-the-box, different point of view. We have a colleague who just came to Hopkins, we recruited, who's an aging expert. And she had shown that fibroblasts from younger people have and this is the stroma, the cells that basically, normal cells that sit in an organ that basically support the function of an organ cell. So let's say you have cells that are making insulin in your pancreas. These are cells that support those cells around there that provide blood coming through and prevent the immune system from killing off normal cells, those sorts of things. Well, what we're finding is, working with her, we've looked at the fibroblasts from normal pancreas of young mice versus old mice, and found that pancreatic cancers grow a lot faster in the old mice with these fibroblasts, with old fibroblasts rather, same cancer cells with old fibroblasts versus young fibroblasts. So again, we don't fully understand this yet, but we're trying to better understand, are there other factors within our bodies that are actually contributing based on age to cancer development and progression. And so this may lead to new ways to look at, why younger people are developing pancreatic cancer versus older people, versus it being only an older person's disease.
Mm-hmm. Thank you for clarifying that. I know both of you have been involved in genetic testing on the GENERATE study, for instance. And I think it's important for us to understand what does surveillance mean? If somebody comes back with a positive and usually a mutation, and usually maybe it's a family member comes back with a positive mutation, what are the steps that they should be taking? I think it would be important for us as a coalition to understand what that protocol is.
I could start, Liz, and please weigh in. So first of all, I think I need to take a step back and emphasize that the NCCN now has universal germline testing recommendations for all patients with pancreatic cancer. And again, this was based on work from several institutions, including Johns Hopkins, Mayo Clinic, Dana-Farber, us, and MD Anderson, showing that if you just use familial history as a criteria to screen patients for germline mutations when they develop cancer, you're going to miss half of the patients who actually carry a germline mutation, but they don't have a clear-cut family history. And so based on that, in, I want to say 2018, the NCCN essentially extended germline testing for all patients diagnosed with pancreatic cancer. Most of our centers now have a pipeline in place where we talk about germline testing with everyone who comes in. And as I just said, you can now get multi-gene testing done for $250. It's a lot more affordable than it was even a few years ago. And so I strongly encourage all the associations on this call to make sure this message gets out. And this is important for a couple of reasons. Number one, for the patient themselves, there are now therapies that can be geared towards certain mutations that these patients may carry. For example, if you carry one of the Lynch syndrome mutations, and this is pioneering work that Liz and others did at Hopkins many years ago, you can give them one of these immunotherapy drugs that has a very good effect in these patients. Or if you have a BRCA mutation, now there are these classes of drugs called PARP inhibitors that can be given to patients carrying those mutations. So there is a benefit to the patient, but there's also an altruistic benefit to the family as well, which is that if they carry a mutation, then the unaffected first-degree relatives, the siblings, the children can be tested and to see if they also carry the same mutation. And again, this is called cascade testing, where you test the patient. If the patient carries a mutation, you cascade it to the family members. Now, if somebody is in fact found to be positive, the first thing to do is to talk to a genetic counselor and not take any decisions yourself, because the lifetime risk of pancreatic cancer may vary, it does vary tremendously based on the exact gene that's defective, or what mutation is impacted, what the family history of other cancers is. Some of these genes, as you can imagine, don't just increase the lifetime risk of pancreatic cancer. There are breast, ovarian, colon, and other cancers also that are impacted potentially. So it's very important to speak to a genetic counselor as part of your care and figure out what needs to be done. And then, as I had mentioned previously, there are, at least in the U.S. and I know that in Europe as well, for sure, there are groups in Germany and many other places that are in the UK for sure that are doing this as well. There are surveillance programs where germline mutation carriers can enroll and undergo surveillance. Typically, I have to say that the consensus criteria for how to do the surveillance and what sort of imaging to use is still a work in progress, even after all these years, and there is some level of disagreement as to what constitutes the best way to do it. But everyone agrees about one thing, which is, yes, if you carry a germline mutation and you are at higher than average risk for getting the disease, you should be in some sort of surveillance programs. And again, this is where a center like Hopkins or MD Anderson, we have specialists who do this for a living and who will tell you, based on this mutation, you can come back, once a year, or you can come back once in 2 years, or you're somebody we think, should have a little bit more aggressive follow-up. So it's very important to be at a center that knows what they're doing in terms of the surveillance program.
But you bring up a really important point, Anirban, is that most people are not treated in a center such as where both of you are at. Most are in community clinics throughout the United States, and I suspect this is probably true somewhat globally too. So
Yes.
I would think it'd be so important, the education of general practitioners. How do we do this? I mean, reaching out to general practitioners has always been very difficult. What's your experience on this? And maybe this is something that we as a coalition, having more information on this could be helpful.
So I'll just say, I agree. You're absolutely right, Barbara. You're bringing up a very important point. I think there's lots of different ways we try to educate, through different organizations about all different cancers. But the reality is, I can't tell you how many times, I hear about someone who's being seen who is told, go home and die. And in fact, we have treatments. And I really do think this is one thing this organization can do, both, in your own country as well as, internationally, to be able to figure out ways to get to practitioners more readily to provide, because the guidelines are there. Anirban mentioned, we have guidelines. They're not reading the guidelines. We have methods, at least in the United States, you have to, every 10 years as an oncologist, you're supposed to be, taking an exam to show you've kept up with all the guidelines. It's beyond me how this can happen.
And Barbara, I will just jump in and say you've been at the forefront of many of this educational outreach efforts for various consortia and NCI efforts. And the outreach and education that you mentioned, it spans the entire disease consortium, from prevention to early detection and interception to treatment. I mean, this relationship between new onset diabetes and pancreatic cancer, I'm always shocked at how few endocrinologists, let alone general practitioners, know this relationship, or the fact that everyone universally should be offered germline testing. It is still a very cachet thing, or actually a niche thing in academic centers. We get patients all the time coming in from the community who were never even told that they should get germline testing done, despite the fact that now we have treatments, as I said, for certain mutations. And so this educational outreach and the organizations on this call have, as Liz mentioned, a tremendous role in that. This information needs to get out to the general practitioners, because that's where the vast majority of patients are seen. And in some countries with national health systems, that's almost exclusively where you're seen until you go to a specialist after a certain point. And
so there is a really good point in the chat. Claudia Mellenthan had said that she's a GP, and she thinks the message that something can actually be done has not reached many of her colleagues. And what she says, which is a good point, we should be involving GPs in the research so that they really feel invested. They get to learn, because that's what research is. We all learn, right? Yeah. And so we're not involving people out there who are in the trenches, helping them to learn in a real way. So thank you for that comment, Claudia.
I think piggybacking on that, the other group that needs always to be involved in research is patient advocates.
Absolutely.
Because we are also in the trenches too, and can bring to a study the patient's perspective, which sometimes is missing, as you pointed out. I want to just turn to something that was exciting that happened recently in early detection. And Anirban, I'm going to have you talk about Immunovia's IMMray PanCan-D test, which has just been introduced and used for screening individuals who are at high risk for developing familial or hereditary pancreatic cancer.
Yeah, that's right. I mean, so we've always talked about not having an early detection test for pancreatic cancer. So finally we have one that is available. It is what is called a lab developed test, or LDT. It is available through Immunovia's site in Massachusetts, but folks can send in blood samples from all around the country otherwise. But the key caveats to remember is that it's not for everyone. And you need an MD to actually order the test. It is geared specifically towards individuals who have a family history, as Barbara said, or one of these germline mutations, and are undergoing surveillance. It's a protein-based assay that I think looks at 9 proteins, including CA19-9, and then based on the results, you get kind of a risk score that goes back to your MD. And again, as with any other blood test, it's very important. By itself, it does not lead to a downstream treatment. It is always looked at in conjunction with imaging data, with other laboratory tests, and then you need to make sure that the physicians talk about this, and discuss what the next best step is. So again, very excited to see that there is a test now available geared towards early detection of pancreas cancer. But the key caveats are: make sure the physicians involved in any decision making don't go out and self-order it, it's for a certain section of at-risk patients for pancreatic cancer, not for everyone.
I would just say to the group here that Immunovia is one of our sponsors, and they have I guess they have a booth or an area, a table. So if you want more information on that during the downtime, please just go over and introduce yourself and ask them more information about this exciting development. We talked about the new onset of diabetes and those studies, and cystic lesions, and chronic pancreatitis is another one of those morbidities. But I want to move from that and talk a little bit about another study that is just starting. And Anirban, I'm going back to you on this too, and then Liz, I'll get your input. It's called the Regenerate study, and this is to understand barriers in the Black community and in the Latinx community about participating in clinical trials. This is just being launched, and it is being supported by PanCan. So Anirban, do you have what to share?
Sure. No, I can give you the background on this, definitely. So Liz and I are actually part of a Stand Up to Cancer grant or team, as they are called, Dream Team, that amongst a lot of different things it did. And I'll come back to one of the things that Liz is doing that is truly pioneering. So one of the things it did was to offer germline testing for high-risk families online through a sort of online-based system where you could essentially consent online, and then you would get an online genetic education, not counseling, but a sort of a pre-test education as to what exactly this involves. And then a test would be mailed to you at home by a company called Color Genomics. And then it's basically a spit kit. You don't even have to give blood. And you would mail it back, and then you would get a test result. And based on that, if you're positive, you'd be given genetic counseling or not. So anyway, so we ran Generate through our initial proposal. And what was disappointing, I mean, it was a huge success. And especially during the pandemic, it was an even greater success than we could envision, because everybody loved not going out to a hospital to get genetic testing done, but having a kit delivered to your living room, right? I mean, what could be more convenient than that when you're in the midst of a pandemic? So accrual was amazing. But what was disappointing to us was that the diversity of accrual was very minimal, in the sense that most of our patients were Caucasian. And so we realized that even when we had, and this is coming back to something, Liz, that you had mentioned, even when we had made germline testing so easy, online, paid for, there were still barriers that existed in terms of increasing the diversity of participants. So clearly, we are not either getting the message out fully, or we are not crossing certain barriers, mistrust maybe of what is exactly being done here, and that we need to sort of overcome. And so to address this, the investigators who ran the Generate study then wrote a follow-up proposal called Regenerate. And the whole idea behind that was to understand, first of all, it's a multi-phase study. The first phase is to understand, well, what are the barriers? Is it mistrust? Is it communication? Is it concern about health insurance, genetic privacy? And so Nicolette Rodriguez and Sapna Singhal and Dana-Farber are leading this effort, and PanCan, very grateful to them for funding it, because we really need to understand. I mean, you can't enhance diversity by saying we're going to enhance diversity. You have to understand what those barriers are, and so this is hopefully one way of getting there.
And I think your point about it's multifactorial. It's not just one barrier. So, and we need to do a better job of understanding, and then we have to come up with solutions of how to really get past those barriers.
Okay.
So I'm gonna jump in one thing and say, so as one of the things that we did in that team, that and Liz, she's being very humble about this, but she was the first one ever to show that in an animal model that you could use a vaccine to prevent progression of pancreatic cancer. So this was about a few years ago that she did this study. And that study was a rationale for proposing what we hope is in fact, the first trial for immune prevention of pancreatic cancer in high-risk patients, really co-opting the immune system with a vaccine and seeing if you can revert or intercept pancreatic cancer in high-risk individuals. So that's another trial that was done as part of this larger team that also did generate.
So a lot of different things going on that are really exciting in this field. A lot of developments. How we talked a little bit, there's a question that I think that we should look at right now that's in the Q&A. And it has to do about Immunovia's test is for one section, one of the pancreatic cancer patients. How near, how far away, what type of research is being done for either a blood test, a saliva test, whatever, in order to look at the larger population?
So I'd like to just bring, and Anirban's clearly the expert on this, and everything he has told you is really state of the art. But I'd like to point out that still in the clinics, we're seeing people who clearly have a family history, but we can't identify that gene or set of genes. And so you could have tests, but if those tests aren't looking for the right thing, they're not going to work, or they're not going to work for enough people who may be at risk, at high risk. And so I think, again, it gets back to we're still at the beginning of understanding, the biology of, and it's a very diverse cancer in a sense, right? Even though it looks to us very similar between patients, we know that the etiologies are very different depending on all the factors we've talked about. But I think what's important now is that the technologies are finally here. Currently when we sequence a tumor, we're only sequencing for like one type of genetic change. There are many types of genetic changes, okay? And so finally, in the past couple of years, we're developing databases of, being able to uncover other kinds of genetic changes. And the more technologies we develop that are that sensitive, the more genes we'll have available to look at, and then the tests will get better. So I'm not sure the tests are bad. I think it's that right now we don't have enough to put into those tests to be able to screen. But I'll let Anirban.
No, you're absolutely right, Liz. I mean, you're absolutely correct. I mean, if you add the numbers up, I mean, and you're an oncologist, so I mean, you see patients, unfortunately, most of them presenting and with no obvious risk factors. I mean, one of the things about pancreas is it's not like cervical cancer where you have one attributable risk factor like HPV. Or lung cancer, where most, not all, but most cases are because of tobacco exposure. In pancreatic cancer, you have that 10% slice of familial cases. You have maybe about 10% from cystic lesions, but you have this 80% piece of the pie where you can't tell the patient what happened. I mean, many of our patients, as you know, and we know individuals like this in all our lives, vegan, athlete, triathlete, and then they end up with pancreatic cancer, and you're looking at them completely helpless because you have no idea what to tell them. Why did you get pancreatic cancer? But clearly there was something that caused it. So slowly we are now understanding the multifactorial basis for why somebody may be at risk. That's not one big thing like a BRCA mutation or a PALB2 mutation. So things like what we call polygenic risk, multiple genes, not having one big effect, but little effects together that add up to a lifetime risk of pancreas cancer. So you take somebody with a high polygenic risk, maybe they smoked, maybe they have high BMI, maybe they also have a family history of, some sort. You mix it all together and it's a combustible mixture. The point is, as physicians, we are only seeing sort of the end product, unfortunately, at the sort of the final 100 meters of the race, 100-meter race. This race began 100 meters ago. If we can identify this process at 20, 30, even 50 meters, there's an opportunity for us to intercept it. But how do we do this, in the realities of the current healthcare system? I mean, I think this is where and Liz is big on this. She, in fact, leads an institute called Convergence Institute. Convergence science is the way to go, which is you got to bring in physicists. You got to bring in folks who study EMR. We complain about electronic health records all the time as physicians because there's so much data to be entered. But that data is still doing something. It is adding information, little nuggets about every individual. If we could somehow figure out what that structured data could be telling us about risk of pancreatic cancer 10 years down the lane, it would be absolutely a game changer. So this is where we need folks who are computational experts, who do AI, machine learning, look at thousands and thousands of medical records, especially some of you who are from national health systems like UK or Western Europe. I mean, we already have those records. How do we mine them to understand who could be at risk with those subtle things, not one big thing like, HPV or a BRCA mutation, but 50 or 100 subtle things that together are adding up to a risk of pancreatic cancer.
So it seems like there's a lot of interest in this area in the last couple of years, developing an AI and in machine learning and applying it to those electronic health records, not just for pancreatic cancer, but in all areas of cancer and other diseases. I know there's some people who are working on Alzheimer's, for instance. So this is something in the future that I'm sure we're going to be hearing a lot more about. Thank you for bringing that up. I would like to skip to something. We just have a few minutes before we're going to then look at some questions here that came in the chat as well as the Q&A. There was recently disappointing data published about the ovarian cancer screening program, which was a really large and closely watched study, and from what I understand, a very well done study. And what implications, of any, does this have for the future of broad-based pancreatic cancer screening?
Did you want me to take that, Barbara?
Please,
Or okay, go ahead. So go ahead. So I think I think you're talking about the CA-125 study showing that there was no impact on survival, and indeed it was. I would say there was a gut check for all of us because we always talk about early detection equating with prolonged survival, but it does also underscore the fact that not all cancers are equal. I mean, I think the data that we have from pancreatic cancer, including from some of these registries where patients have been able to undergo early detection and early intervention and undergo surgery, many of those patients are cured. If you look at the world of long-term survivors in pancreatic cancer, and thank goodness, every year those numbers keep going up. Yes. Many of us who've been to PanCan events, Julie always asks long-term up, and there was to be one or two individuals. Now we have a room full. It tells you that our therapies are improving. Patients are getting longer term benefits, but there is a catch. That catch is these are all individuals who were diagnosed early in the disease, were able to get to our surgeons and get the tumor taken out. Metastatic tumors, unfortunately, still is a tough disease to deal with, and unfortunately the biggest piece of the pancreas cancer pie. And so if we can get more of our patients to an earlier stage of diagnosis, get patients to our surgeons, give them adjuvant therapy, and maybe the vaccines that Liz and others are making in the maintenance setting after that to really sort of clean the clock, so to speak, and get rid of even any smaller cells. I think that we are really going to have an impact on survival. I mean, I think that what may have happened in this one study in ovarian cancer, that is a reality check, but doesn't necessarily in any way, to me, avert the need for continuing early detection and interception efforts in pancreatic cancer.
Liz, can you comment on, and Anirban just brought this up, about the vaccine? Can you tell us a little bit about this and how long you think that this would actually be something in a clinic?
Yeah, I don't, well, it should be in the clinic this year to begin testing just as a proof of concept and safety, of course. This is a vaccine we did test in patients who underwent surgery, who have no evidence of disease. Like Anirban was saying, they're still at very high risk for recurrence. And so we just started that. Believe it or not, we got approved January of this year. We've already enrolled 6 patients and have data on the patients who have undergone surgery and have now taken this vaccine, and we know we can induce a good immune response. We don't know what the immune response means yet because it's only been at most 6 months for these patients who started on this vaccine therapy in that setting, but it's safe enough that we felt we could go to the FDA and get approval to test it in high-risk patients. And so Anirban and some of my colleagues here, Mike Goggin, both who lead the Stand Up to Cancer grant that we participate in, have come up with the groups that we should test this in the clinics where we screen high-risk patients and families with pancreatic cancer in their families. And so we basically will be, all the ones we've mentioned, it's the families with a history of these cysts, BRCA, PALB2, ATM mutations. These are the groups that we're going to offer this vaccine that we truly believe is safe. It targets mutated KRAS. And we at least want to see if we can induce good immune responses and try to understand those immune responses, what's important, and then follow these patients longitudinally. So it's going to be a while, but, like Anirban said, I mean, this is a big advance because up until now we've been in mice and we've proved it in mice many times, but now we really have to take it to the people who need this. And safety was the primary reason we waited, but we believe it's safe.
So it's a really exciting step.
I think so.
Yeah.
I think it is.
Yeah.
But it's a first step. It's a baby step. I don't want to, tell people we have the answer, but we're learning. I'm happy to share as we learn. The more participants, the better. So we're very, excited about this.
We look forward to hearing more about this. I'm going to now go to the Q&A, and I see there's a lot in the chat too, so I'll go back and forth between the two. But there's a really good question here. Is there overlap with all these similar studies happening? There seems to be some studies that overlap of the same patients in different studies.
Yeah, I mean, I think, just talking about the risk factors, I think I mentioned this previously. Many of these genes, germline mutations, are not pancreas cancer exclusive. They also increase the risk of other cancers. And in fact, sometimes the other cancers are dominant cancers, like in Lynch syndrome. Colon and endometrial are the dominant cancers, but there's also risk of pancreatic. In ATM, for example, breast cancers are often the more common cancer. And so again, this is why it's so important that one speaks to a genetic counselor if you receive a positive germline mutation diagnosis, because a genetic counselor will be able to walk you through what the risks are, not just for the pancreas, but other cancers, and how other family members should also evaluate their risk, etc. So it's very important. And yes, there is overlap between some of these studies.
Okay. And is that a negative that there's overlap, or does it help support?
No, I mean, I mean, Liz and I, we talk about this, I mean, I think lessons learned from one cancer type are very important in the other cancer type as well, right? I mean, sometimes many times they're different, but you still learn lessons from them, right? I mean, what you learn from prostate cancer may or may not be applicable to pancreatic, but there are lessons you still learn from prostate cancer studies that you can at least co-opt.
Right. Liz, this is a question for you. How much research is actually dedicated to prevention that you were aware of?
Yeah, that's a great question. There was a time in the 90s where chemo prevention was looked at, particularly in breast cancer, and unfortunately, basically manipulating estrogen receptor therapy, it turned out it wasn't worth it to women at high risk. The side effects were worse than the risk of getting breast cancer because you could screen for it. And then I think there was like this lull. And now, prevention was, again, a very high priority that we came up with at the Blue Ribbon panel. And the NCI has really put a lot of effort lately into supporting prevention. And I just actually helped co-organize a workshop that the NCI had last week. And I think these workshops are very important because they bring people from all over the world together to come up with new ideas, and then it generates more funding. So I think it's getting to be a higher priority, but it's something that we should continue, because prevention to me is really where it's at. We need early detection because that allows interception, preventing the cancer itself. And then hopefully we could even prevent an early lesion eventually. So those are the big goals.
Mm-hmm.
An ounce of prevention is worth a pound of cure, right? Benjamin Franklin said that.
Yeah, but it's great to hear that the NCI is in making this a global effort, not just egocentric for the United States, but that it's including researchers from all over the world. It gives us a better opportunity on that. Let's see. Are most of the studies indicative of the disease? Are the demographics indicative of the disease per the percent of race and sex that is indicative of the disease? I think what this question is about, are there any differences in the demographics in the disease on race and sex?
So I think it's slightly elevated in African Americans, not like prostate or breast cancer, but certainly it is. And then also men are slightly at increased risk over women. And again, I don't know if these are risk factors in the environment versus genetic risk factors. We don't know a lot about why there are that slight differences.
Yeah. Okay. But there are definitely ethnic differences. I mean, there's studies from, there's a cohort called the multi-ethnic cohort, or MEX in the US that specifically focused on collecting individual samples and histories on patients from diverse ethnicities. And so, as Liz said, African Americans have a higher, not very high, but somewhat higher risk than Caucasians. But I think the highest risk is actually in Hawaiian Americans, just putting it out there. The study, particularly this study, found that the risk is several-fold higher in Hawaiian Americans than it is in anyone else, in all of these other ancestries. Again, the reason is not known, probably something genetic. Who knows? There's also, these days we also worry about the microbiome and other environmental factors. So we don't know why, but there are certain ethnic groups where the risks are much higher. I mean, but this is true for any disease. Like, we know, for example, in Native Americans, diabetes rates are very high. Latin Americans, in Latino communities here in Texas, for example, diabetes rates are very high. Much of that is environmental, but some of it is obviously genetic as well.
So here's another question that we touched on a little bit, but I think because it's been upvoted so many times, I think we go back to it. The ultimate prevention mechanism will be a blood test to detect pancreatic cancer earlier. I suspect some type of test, whether it's blood or urine, saliva. Please comment on recent developments in developing such a test. And I suspect that this person is writing about sporadic cancer and not just in the familial genetic area.
Yeah, but that's a great question. Liz, do you mind if I talk about it? Okay, so I think there's a lot of interest, obviously, has been for a long time in developing early detection tests for pancreatic cancer. I already mentioned the fact that there is concern about screening everyone, and so screening and surveillance should really be restricted to high risk, but that still doesn't exclude the development of these tests, and there's a lot of activity in that space, obviously. So I talked about the Immunovia test, which is, again, for a very defined population. But then there are also other companies that have been developing tests that are focused on circulating abnormal DNA, for example, in the blood, a method that just falls under the umbrella of something called liquid biopsies. And there's been a lot of interest in liquid biopsies for early detection. Again, not to endorse any company or anything like that. There's a company called Grail that has developed a test called Galleri. Galleri is available in the US, and PanCan, Alison Rosenweit, had a very nice blog on both Grail and Immunovia's test. So Galleri is available in the US. Again, it's not FDA approved. It is, but you can order it if your physician orders it, and it's an out-of-pocket payment that has to be made. And that basically looks for snips of abnormal DNA that's circulating in the blood that could be released by a cancer. And it's not just for pancreas cancer. It detects any multiple types of solid tumors, and also looks for information of where that tumor might be coming from. So the pattern you see in the blood will be different for pancreas versus lung versus breast. And so this Galleri test is available in the US, costs about $1,000 out of pocket. So it is expensive. But there's a trial going on in the UK. The NHS and Grail have partnered together to actually have a trial where, again, individuals who are above 50 can be tested using this test as part of a clinical trial. And then there are other similar assays available. One that was actually developed by one of Liz's colleagues, Bert Vogelstein at Johns Hopkins, and then it was licensed to a company. And that's also, it's undergoing testing right now. It's not commercially available like Grail is. And then there's a slew of companies that are looking at blood tests for early detection. Again, it's a challenging area. Yes. because you have to show that you are able to detect cancer earlier, and that early detection translates into improved survival. So there's two levels of bars that need to be met here. The earlier detection of cancer is what we call a stage shift. Instead of 80% advanced stage cancers, you're now getting only 50% because more early stage cancers have been detected. But that's one bar. The second bar is also needed, which is how does that translate into better survival? And so this is a complicated and difficult path. One of the reasons why we don't have a lot of approved tests, so to speak. But that doesn't mean that there aren't a lot of players in this field that are very interested.
Mm-hmm. Yeah. There was a request to share data and evidence about the issue of diversity and the inequity of pancreatic cancer care and treatment. I think we can take care of that offline. Jenny, later, if you work with me on that, we do have stats on that we could share. What can you suggest in carriers of mutations of BRCA1 to PALB2 that lead to genetic predisposition for breast, pancreatic, prostate cancer? So you've talked about surveillance and a genetic counselor. Are there any other ideas that you want to add on to that?
Yeah, I think, again, it depends a lot. This is an international audience, Liz, and so please add on here. But I think, some of it depends on what's going on in the country that somebody's identified. Like in a place like India, for example, where I was born, I don't know what surveillance program exists, if any. And so it all also boils down to access and what's available. I can tell you what I would do if you were at MD Anderson or Johns Hopkins, but that may not translate into an answer that equates everywhere else. So, but at the very least, if you can speak to your physician and genetic counselor and figure out what the next steps could be.
And my understanding is that genetic counselors are now doing virtual sessions, and so that should be helpful somewhat for people who don't have access at their local community center.
Yeah, absolutely. One of the things that the pandemic did, which unfortunately we're seeing now being pulled back, and we're hoping it stays, is it really made telemedicine very easy across state lines. And I think patients love that. Especially patients with cancer who are unable to travel. Imagine being able to have a consultation from your room. And I think that convenience hopefully will be a transformative change that stays even when the pandemic is hopefully gone soon. So
yeah, there are a lot of challenges. We're losing that ability in many ways because of how physicians are regulated in different states. But also it doesn't help people outside of the country. Health, because you still have to pay for it. And so poor people are still not going to be able to pay for it. And I'm sure health insurance in India is not going to pay for them to get consultation in the United States. It's a huge problem. We have to solve this. This is bad.
Should all patients diagnosed with cysts be followed up?
Again, the guidelines vary. You'd be amazed at the heterogeneity of guidelines by different organizations. There's a whole slew of them. But overall, the management will depend on several things: the age of the patient, are there other complicating or mitigating factors? Where is the cyst located? If some of these cysts, if they're in the main pancreatic duct, or the patient is having symptoms, will immediately undergo surgery. Mm-hmm. But other cysts that are in what are called branch ducts, in a tributary of the main duct, they may be followed over time with imaging. The intensity of imaging will vary. Sometimes it's every 6 months, sometimes every once a year, again, depending on whether there are other things that the surgeon or the gastroenterologist sees that concerns them about the cyst. So it's a very heterogeneous answer, so to speak, because it is a very heterogeneous field.
Yeah.
And when to even stop screening? Some societies say stop screening at 5 years if nothing has happened. Other societies say, oh no, you need to screen for the rest of the life because the pancreas is at risk. So I guess there's a lot we don't know is the simple answer to your question. So, and this is again why I think it'll be great to have some interception things that don't involve surgery, Liz. I mean, we could give a vaccine against an IPMN, or something oral that the patient could take, like a pill a day or a vaccine.
That'd be even better, sure.
There's a question here, Liz, I'm going to give you this one. And it has to do with how do you help someone emotionally when they have pancreatic cancer? And I know this, you're an oncologist, so you have to obviously deal with this a lot in helping and supporting patients and their families. So what would you suggest in that?
Well, the first thing gets at what we've been talking about. And, a lot of patients that at least get to me and they get my email and they email me, and I'll be reading this on Tuesday night at 10 p.m. And they were told their family member has pancreatic cancer. They were told to go home and die. And I respond and I say, well, I need to know more, but, we do have research going on. And, today research means something different than it did even 5 or 10 years ago. Today, some of those agents that are in clinical trials are actually working. And we're seeing patients, even pancreatic cancer patients, live years instead of just a few months. Now, not everybody. We want everyone to be there. But by responding, by giving them hope, by giving them options, we all want options. We know that the answer is not going to be, oh, I can cure you. They know that by then. But they need options. And many of the people who I interact with in those situations, even if I end up not being able to help them, they felt like at least somebody was there who knew something, who wanted to try and give them options. And so I guess that's my way of doing things. I want to, and that's why I'm in this field. That's why I just, I needed to be trying to find the right answers to this disease. Of course, I may retire and not be able to, but hopefully other people will, like Anirban and everybody else who's working on it. I hope I'll still be a part of this. But to me, that's the most important part, is giving people hope, giving them options, and knowing that we're making progress. And I think that's important. And they, they become a part of the team. They're helping us do this. And I think that's important. You need to empower patients and their advocates to be a part of the team. You have so little control when you have cancer. So little control. You need some control, and taking control and being able to be a part of the team to help find the answer, to try things that may work. I think, to me, that's what I can offer someone the most. Mm-hmm. And that's how I look at it.
Mm-hmm. And then helping them with the decision making. Yeah.
Of course.
Anirban, any other thoughts on?
I mean, Liz really summarized it well. I mean, and this is, I think, one of the reasons why this alliance is so important, because you have a finger in the community that, physicians may or may not have. And so, you may be the first sort of outreach that a patient makes.
Yeah.
So hopefully, the things we talked about and some of the optimism we talked about will be carried back by you to the communities that you interact with.
I have one last question before we sort of wrap up here. There's a lot of talk about out-of-the-box ideas for early detection. Some examples are wearables, such as self-monitoring watch. There's the eelworms to detect cancer cells in urine, dogs who are trained to sniff for cancer-related scents in patients, an intelligent vase toilet that monitors human waste, and use of social media, specifically monitoring searches for symptoms, which is developed into its own area of science called social ethnography. Now, I'm only bringing this up because this is all in the popular press. And so, I don't know, Anirban, do you want to start on this? What are your thoughts on these approaches? Are any ethical dilemmas that need to be involved here?
Well, I mean, I think I don't know who's going to win between the urine sniffing eel and the urine sniffing dog.
Yeah, I'm not sure.
Yeah, I don't know, but I mean, you there's a lot that's reported in the press. I mean, you have to be careful because, again, sometimes companies will splash something based on very few patients or a small number, and you have to be a little bit careful about, and that's why I think, again, organizations that are on this call are so important, because sometimes you are the gatekeepers of good information versus misinformation. If there's one thing the pandemic has taught us, there's a ton of misinformation online. And so again, we look to you to some extent to be guardians of at least the pancreas cancer part of good information. And I think I know that many of the component organizations reach out to me sometimes saying, "What do you think of this? Is this real, or is this just a sort of a hype?" And so again, you have to be careful to sort through the data. Is it published? What does the raw data show? Is it just hype? I'm happy to do that, and I'm sure many of us are, and PanCan and Love's Garden and Let's Fit and others do a fantastic job of sort of addressing some of these questions in written blogs or press releases of their own, etc. So I think there's a lot happening, but you just have to be wary of what's real versus what's hype. So I'll just.
Thank you. So it is our privilege to represent others, both the voices of patients as well as translating the language of science, advocating to government, raising awareness in our communities, raising funds for research and education, donating or volunteering. In addition, it is important that we as advocacy groups understand the role we play in motivating, empowering, and supporting change on behalf of all stakeholders. I'd just like to, as a final message from both of you, what role do you see the members of the World Pancreatic Cancer Coalition play in changing the outcomes globally for pancreatic cancer?
Please.
I'm happy to start. I think, clearly we have to do a better job of educating the public. And there are so many different ways, different social medias, different ways to do this. And maybe we have to do all of them. Maybe we have to look at each country or each, section of a country and figure out what is the best way to get it out there. And we have to get it out to people. We have to get it out to caregivers. We have to get it out to, physicians, to nurses. So we need to do a better job of getting the right messages out to people. I'm not saying I know that way, what the best way is. And I look to all of you because you're doing more of that in many ways than I am, but tell me what I can do to help. And then collaboration on clinical trials. And this is very important because, there is a great disparity of access, whether it's in places in the US or if it's in countries all over the world. I mean, I get calls and emails from all over the world, from all over the country. And I'm really, it's hard to be able to offer, even when we have clinical trials, to these individuals. And I think we need to do a better job having governments and industry work together to be able to allow patients to access trials, because it requires money. It requires good health care. And those are the two places where we get funded. We get funded from industry, we get funded from the government, whether it's healthcare or healthcare companies as well. In the US, a lot of them are private. But that's how we do our research. And so I want to be able to provide this to everyone. So we have to figure out how to lobby these people and get them to realize cancer is a real problem. Many different cancers. Pancreatic cancer is deadly. And we have to work together to do a better job. And then we have to incentivize collaborations on sharing data. I think what we've learned from COVID is that in 1 year, we can come up with a vaccine that can save lives. Yeah. How did we do that? Well, we did that because we had good science that we drew upon. Well, we have good science in pancreatic cancer. A lot of money was put into it, yes, but very importantly, all of a sudden, every morning I would open up the newspapers, journals. There was all this information that was free. I didn't have to pay to get that information. Now, yes, I have access to lots of journals here in the US, but imagine being able to share this information between countries, between all sorts of people. That's how we made progress fast. We need to be able to do this more. And I have to say, and I'm ashamed of some of the groups I've been a part of, but we don't do that because why? It's not that people don't care or groups don't care. But for instance, a lot of groups make money off of their journals, and that's how those groups are supported. That's one example. But I'm sorry, data has to be out there and free and accessible and usable. And the other group that's really tough to share with is companies. They're sitting on lots of data and it's proprietary. And we don't know how people are responding to all these different drugs. Anirban and I can't make the next big discovery without seeing all that data. So I think, lobby to get people to share data.
It's a good point to end on. I want to thank you both. And Anirban, did you have anything?
I have nothing to add. I cannot up what Liz just said.
You said it brilliantly, Liz. Thank
We can
you.
all clap to what she said, because that's
Well, I think everybody should clap for both of you. You were just
Yes.
so wonderful to come today, share your knowledge and expertise. There was a lot of thought-provoking ideas and a very informative session. So thank you.
Thank you.
And now.